first step is called G1 of interphase, or growth 1 of interphase. As you can see, we have 8 chromosomes within our cell because there are 8 different lines or 8 different chromatids. Proteins are coming into the cell, adding to its mass, making it larger, that's where growth comes from. Each of the 8 chromosomes is assigned from each one of the parents, mom and dad. There are 4 chromosomes from the dad and 4 chromosomes from the mom. Dad will be blue and mom will be pink. Because of this also means that there are 4 homologous pairs of chromosomes. The next step of interphase is S of interphase, or synthesis of interphase. As we can see up here, there are duplicated chromosomes, meaning the previous chromatids, they are now doubled, so it is in an X shape. However, this does not mean there are more chromosomes, it just means there is more DNA. So as you can see here, capital C, this is what the line means, capital C duplicates. It still is capital C, even though it duplicated. So this is still 1 chromosome. So there are still only 8 chromosomes. The next step is G2 of interphase, or growth 2 of interphase. During this step, the cell intakes living mass to increase its size. As you can see, it is visibly larger since S, or synthesis of interphase. And as you can see, the proteins are entering and creating a larger cell for the chromosomes. This will eventually lead to the dividing of the nucleus. We are now moving on from interphase. The next step is called prophase 1. In this step, duplicated homologous chromosomes are paired together with one another, one from the mom and one from the dad. Once they are paired together or embracing, they are called tetrads. These tetrads exchange pieces of genes from each other and to each other. As you can see, D is giving to D and A is giving to A. It just exchanges. This leads to genetic diversity among chromosomes and eventually among actual organisms. You can also tell that the membrane of the nucleus is starting to dissipate. The next step is called metaphase 1. Homologous pairs line up randomly each time meiosis occurs. This is called independent assortment. As you can see, we have 4 chromosomes on this side and 4 chromosomes on this side. Also, this is the result of crossing over. This independent assortment causes variation among chromosome distribution. After this, the spindle fibers attach to the chromosomes. As seen in metaphase, the fibers attach to the chromosomes. However, this step is anaphase 1. In this step, the fibers pull the chromosomes away from the middle towards the opposite ends of the cell. The next step is called telophase 1. After the spindle fibers pull each homologous pair to their side, they dissipate. After they dissipate, nuclear envelopes begin to form around the pairs of chromosomes, the homologous pairs of chromosomes. So this one forms around this 4, this one forms around this 4, and as you can see, the cell starts to morph as if it is going to split. The next step is called cytokinesis 1. As you can see, 2 whole cells have formed. However, these are haploid cells. As seen, the starting cell had 8 total chromosomes, but these haploid cells only have half the number of chromosomes, which is 4. This is called reduction because we are reducing the total number of chromosomes from the beginning. So each one has exactly 4 duplicated chromosomes. In a real-life scenario with a human, it would be 23 each, which are haploid of the 46 that the original cell starts with. The next step is prophase 2. As before, we now have 2 complete cells, or not complete if you would call them half. So 2 complete haploid cells, and they are now about to undergo metaphase 2. The next step is metaphase 2. In metaphase 2, the chromosomes line up in the middle as in metaphase 1 of each cell. However, they are not homologous pairs, but they do independent assortment because the chromosomes are randomly lined up in the middle of each cell. The next step is anaphase 2, which is where things start to get a little more different. In each cell, instead of pulling the pairs, since there are none, they actually split the chromosomes, so they pull the chromatids apart to each opposite end of the cell, and this goes for both cells. So they pull the chromatids apart, essentially splitting the duplicated chromosome in half. The next step is telophase 2. With the chromatids on each opposite end of each cell, nuclear membranes start to form around them. This will start forming 4 new cells, 2 from each. The chromosomes then decondense. The next and final step of meiosis is cytokinesis 2. In cytokinesis 2, we can see 4 individual cells with 4 chromosomes each. As we can see, 4 chromatids, 4 shapes, so 4 chromosomes. That goes for each of the 4 cells. Each cell is haploid, meaning it has half of the original cell, 4 and 8. 8 for the original. This is the final product of meiosis.